Hello, my name is Sean Schaefer. I'm the Genome Editing Platform Lead at Aldevron, and I'm here to talk to you today about the manufacturing of CRISPR RNPs for clinical applications. One of the things I was drawn to Aldevron for when I began there a few months ago was their ability to supply GMP-grade Cas9 proteins for clinical applications. In the course of working with our therapeutic partners, we came to find that there was a similar need for clinical-grade RNPs, and so we began a course of work to identify the best way to complex and characterize these RNPs, and that's the data I'll be presenting today. The CRISPR RNP is the ribonucleoprotein and consists of the guide RNA complexed with the Cas9 protein. In this silhouette here, you see the Cas9 protein outlined in the background. It's complexed with the guide RNA as it associates with the genomic DNA. And once the guide RNA is aligned with the complementary genomic DNA sequence, these two little claw arms silhouetted here representing the catalytic nuclease domains, cleave the genomic DNA, creating a double-stranded break. And this is the process that researchers use to inactivate or knock out a gene. So when we think about relevant examples of CRISPR RNPs being used in clinical scenarios today, the first thing that comes to mind are ex vivo applications for CAR T and TCR cell generation. So in the last couple of years, the first CAR T therapies in the forms of Kymriah and Yescarta were approved, and these simply consist of a carcass set delivered by a lentivirus randomly integrated into the T cell genome. In the intervening years, quite a bit of research has been done to show that inactivating endogenous T cell genes can lead to an improved therapeutic profile for these immune cells. And in the course of this work, it was found that CRISPR RNPs are the best way to accomplish this. So CRISPR RNPs are immediately active. They circumvent the need for transcription and translation. And importantly, they're possibly the most drug-like delivery format when we think about CRISPR formats of plasmid and mRNA, because again, not needing transcription and translation, they are much more like a small molecule. And in that way, they're much more titratable. And importantly, we can come up with in vitro assays to characterize their performance and their composition. So at the bottom here is a table showing some of the T cell genes that have been shown to improve efficacy of therapeutic T cell profiles when knocked out. So the endogenous T cell receptor genes, when they're knocked out, can prevent cross-reactivity and end up with a singularly targeted T cell to a specific cancer antigen. The PD-1 gene that when knocked out resists T cell exhaustion, and then HLA genes, which are responsible for initiating the graft versus host disease response. And so when we think about making off-the-shelf or universal allogeneic T cells that will work for everyone, knocking out the HLA genes is an important consideration. And so these are just some of the most recent examples of ex vivo RNP applications using CRISPR. So as I said, Aldevron was approached by our therapeutic partners who identified a need for characterizing CRISPR RNPs under GMP conditions. And so we set out on this project to do exactly that. Our project goals were first to identify the ideal complexing conditions for guide RNA and Cas9 proteins, to create an in vivo cutting assay that would be platformable from one RNP to another, to determine the best way to identify free Cas9 and sgRNA in the RNP composition, to create a standard QC panel for RNPs, and then importantly, to conduct long-term stability studies for RNP, because again, if we want to think of these as an off-the-shelf drug, it's important to understand their storage conditions and longevity. And a lot of this work culminated in this poster you see at the left here, but is also ongoing, and we look forward to continuing to develop this field in the future. But let's review the data that we have so far. So we begin with the development of an in vitro cutting assay. And so this is an important way to, again, characterize RNPs in a drug-like fashion in an in vitro and very replicable setting. And so in the top left gel here, you see diminishing concentrations of RNP with the highest concentration on the left. And for those of you not familiar with looking at these gel-based cutting assays, in the first lane to the right of the ladder, the top band there represents a linearized 5 kb piece of plasmid DNA that contains the guide RNA target. When the guide RNA binds and cleaves in the RNP composition, it creates a 2 kb and a 3 kb band. And so when you see robust cutting taking place, you see diminishment of the top band and an increased density of the bottom two bands. So as you might expect, at the highest concentrations of RNP, and I'll add that these were done at a 1-to-1 guide to protein ratio, we see robust cutting and almost a complete abolition of the top band. And as the RNP concentration decreases, we see the top band growing brighter and the bottom two bands disappearing. And so what we wanted to do here with creating this in vitro assay, it was important to be able to define a linear range of cutting as well as the limit of detection for RNP activity. And so as you see in the graph on the left here, we were able to establish this nice linear range of cutting activity, as well as a limit of detection of around 0.01 picomoles of RNP. And so we had a very nice in vitro assay, and there's more data with different RNPs supporting this, but this is a good representation of this assay. Now that we had a reliable in vitro cutting assay, we then moved to looking at the ideal ratios of guide RNA to Cas9. And again, this was all done in vitro, and it was substantiated and backed by cellular cutting activity that won't be presented here. But as we look at the bottom left graph, this is the same concept as the top graph, linearized 5 kb plasmid that has the target for the guide RNA within it. And so the first 4 bars to the right of the ladder here represent, and importantly here is the non-target control, or rather the negative RNP control in the second lane. And so you see only the top 5 kb band here and no cut bands below it. But as we look at the first 4 lanes to the right of the ladder, we see here, this is the 0.5 guide RNA to 1 protein ratio. And you see cutting, again, at the highest concentration in the 4th lane from the left, indicating that cutting is taking place, but not very robustly. As we move to the right, the guide RNA to protein ratio increases from 1 to 1 to 1.2 to 1.5 to 1 at the highest ratio. And as we look to the far right 4 bands, we see that at the 1.5 to 1 guide RNA to protein ratio, we see the most robust cutting and almost a complete abolition of the target template DNA. And so this is an important finding, as I mentioned, that was substantiated with cellular activity as well. And when we think about clinical assays, we think about GMP formats of protein and guide RNA. These are fairly precious and expensive reagents. And so it's really important to find that optimal ratio of guide RNA to protein and not any more or any less than is necessary. And so again, this was an important finding looking at the ways to optimize guide RNA to Cas9 protein ratios in vitro. We then found it was important to look at the composition of the reagents and look and see how much RNP was there versus how much free Cas9 and guide RNA was there, again, especially if we want to look at this in a drug-like way. And so we started off looking at a couple of different assays, beginning with differential scanning calorimetry, and then moving on to size exclusion chromatography to assay the amount of free Cas9 in the solution. And so as you see on the left here with the differential scanning calorimetry, we were not able to find a linear range for titrating free Cas9. And so we moved on from this assay and then investigated the ability of size exclusion chromatography to serve this role. But as you see here on the graph on the right for the RNP SEC, the Cas9 represented by the blue bar here elutes right on top of the RNP with the guide RNA eluting slightly later. And so this size exclusion chromatography was not found to be a reliable way to differentiate between guide RNA bound and unbound Cas9. We then moved on to analytical cation exchange as a method for assaying free Cas9. And so looking at the graph on the far left, this is the titration of Cas9 all by itself on an analytical cation exchange column. And you see that free Cas9, or rather unbound Cas9, elutes just after 15 minutes on this column. When we look at the next graph on the right, this is RNP on the same analytical cation exchange, and it elutes between 9 and 12 minutes. And so when combined together on the graph on the right, you can see that you get nice separation of the elution times between RNP and free Cas9 represented in the blue peak on the far right. And so after quite a bit of optimization of conditions, we settled on analytical cation exchange as a reliable method for assaying the amount of free Cas9 in an RNP composition. We then, it's worth noting at this point that the quality of the guide RNA can have a substantial impact on the reliability of the analytic cation exchange assay. And so as an example here, on the gel image on the left, in the first lane next to the ladder, you see a high quality, highly purified guide RNA. And in the adjacent 5 lanes, you see a lower level of purification guide RNAs. When we then run these as RNPs over the analytical cation exchange column, on the top right graph, you see now we again have nice clean separation, 10 minutes of elution time for the RNP, around 15 minutes for the free Cas9. And so again, we're able to cleanly distinguish and quantitate the amount of each in the composition. When we run the same assay with the lower purity guide RNA, you see now that there is no separation between the free Cas9 and the RNP, and so making this assay unreliable. And so we thought this was, again, important information for not just our therapeutic customers, but also research customers to understand that the qualities of the guide RNA can have an impact not only on RNP composition, but also on RNP quality and activity. We then turned our attention to being able to also measure the amount of free guide RNA in the RNP composition. And so across the top here, these are increasing ratios of guide RNA to Cas9 protein. The left represents a native page gel run with the RNP, and here down at the bottom, you see the unbound guide RNA. And as you might expect, as the ratio of guide RNA to Cas9 protein increases, so does the amount of observed free guide RNA at the bottom of the gel here. To confirm that this is indeed free guide RNA, we ran a Western blot on the same native page gel using an antibody against Cas9 protein. And as you can see here on the right, these red lanes represent the staining for Cas9 protein and the RNP complex, confirming that it is indeed unbound guide RNA at the bottom of the gel here. And so we found this to be a reliable way to distinguish the amount of free guide RNA in the RNP composition. As I mentioned, if we want to treat CRISPR RNPs as a drug where we can store them and pull them out of the freezer as needed, it's really important to understand the long-term storage conditions for CRISPR RNPs. And a search of the literature finds that there is actually very little data around this. In general, the guide RNA and the Cas9 protein are complex just before application to the cells. And so there is really an absence of data around optimal storage conditions and longevity. So Aldevron began a project to identify these conditions. And so we looked at storage at -80, -20, 4 degrees, and 30 degrees for up to 6 months, and then at -80 degrees for up to 24 months. When we look at our 6-month data, and so these gels are the exact same cutting gels that we looked at earlier with a 5 kb plasmid containing the RNP target and the 2 bands below representing cut DNA. And so the top right gel here, next to between the ladders, we have -80, again, decreasing concentrations of RNP, and then the exact same assay run at -20, 4 degrees, and 30 degrees. And so when we look at the first lane for -80 next to the ladder here, we see that robust cutting is taking place of the template DNA, very similar to what was observed at time point 0. We see similar cutting taking place at 20 degrees and 4 degrees, but then when we look over here at 30 degrees storage, we see that the cutting has diminished. And when we move from the highest to the second highest concentration, we see that cutting is actually substantially lower than what we see at the other storage conditions. And this is probably not surprising, right, that storing protein and RNA at high temperatures results in degradation. And so, but it is also encouraging to confirm that storage at colder temperatures seems to preserve the RNP's activity and composition. And again, this is borne out by our analytical cation exchange assays. And so these are a high guide RNA to protein ratio of 1.5 to 1. And so you see here very little free Cas9 over here at 15 minute solution, and nice robust peaks representing fully formed RNP, very similar to what we observe at time point 0. When we move over to -20 degrees, we see a nearly identical peak representing RNP and a similar peak at 4 degrees. However, as might be inferred from the cutting activity data, when we look at the analytical cation exchange results for 30 degree storage, we see almost a complete abolishment of fully formed RNP, indicating that degradation is likely taking place. We then extended these assays out to 12 months, as I said, only at -80 degree storage. And so here, side by side at the top, 2 gels are the 6 months data and the 12 months data. And so again, when we look at the top band, what we want to see is comparability, and indeed we do, that there is absolutely comparable cutting taking place at 6 and 12 months time points when stored at minus 80 degrees. And then we look at the bottom two analytical cation exchange results. Again, we see very comparable peaks corresponding to fully formed RNP and little degradation products. So again, very encouraging that up to at least a year at this point, we see robust ability of the RNP to cut and show few degradation products when stored at minus 80. We then looked at free guide RNA, and here, so on the top gel is 3 months results, and this again represents storage at minus 80, minus 24 degrees and 30 degrees, and then the bottom is the 12 months results for free guide RNA, only at minus 80. And so the first 7 lanes represent a decreasing titration of guide RNA. This is again the native page gel. And the 4 right lanes represent the 4 storage conditions. So the fourth lane from the right represents minus 80 degrees. And so when we look at the free guide RNA standard, we see our free guide RNA here, and it looks very nice, a very nice tight band, few degradation products below. We see a similar band at minus 20 degrees, but then when we move to 4 degrees at 3 months, we start to see degradation products. As we know, RNA is fairly labile, and so maybe not surprising that we start to see degradation in a fairly short period of time, even though, as I'll point out, we saw strong activity in RNP complexing at these same storage temperatures. And then, again, maybe unsurprisingly, when we look at 30 degrees, we see that the free guide RNA is gone, indicating that it has likely been fully degraded. And so, again, these are probably not surprising, but great confirmation that at least at minus 80 degrees and minus 20 thus far, we see robust and optimal storage of the RNP. When we then move down to the bottom gel, this is minus 80 results only at 12 months. And so importantly, we see again this nice tight band. These are triplicates of storage at minus 80 and few degradation products below or above, indicating again that at least up to 12 months storage of the RNP at minus 80 degrees is a reliable way to contain these things for a long period of time. And so we, as I said, are conducting these experiments out to 2 years and look forward to reporting that data. So I hope that you've enjoyed this talk. I'm happy to take any questions that you might have if you're interested in using CRISPR proteins or RNP for your research or clinical use. And I thank you for your time.